In the last lecture for this week, I'm going to discuss with you the different types of receptors that can be found on the cell surface that can lead to signal transduction within the cell that will alter the cell's behavior. And the types of receptors fall into three broad categories, which we'll get into. The first category of receptors you're already somewhat familiar with because we did talk about this category of receptors when we discussed the different types of proteins that can be embedded in the plasma membrane. And this type of receptor is the ligand-gated ion channel. Ligand-gated ion channels go by a few different names. Your textbook also refers to them as ion channel coupled receptors. And you may also see them listed as transmitter-gated ion channels because sometimes the ligand is specifically a neurotransmitter. So all three of these mean the same thing and they essentially function in the same way. So we know what an ion channel is. An ion channel is a membrane protein that allows ions to enter or exit the cell. When an ion channel is ligand-gated, that means it exists in a closed and an open state. And when the ligand is not bound to the ion channel, the channel is closed and the ions cannot pass through the channel. However, upon ligand binding to the channel then opens and now ions can pass through the channel. So the ligand binding serves as a way to open the gate, so to speak, to allow the ions to pass through. When the ligand is not there, the gate is shut and the ions cannot pass through. The second type of receptor is something very different that you may or may not have ever heard of before, and these are the G protein coupled receptors, or GPCRs for short. G protein coupled receptors are very large receptors. They span the plasma membrane 7 times. So if you look at the basic structure of a G protein coupled receptor as shown here on the slide, if you count how many times that receptor passes the plasma membrane, you'll notice it spans the membrane 7 times. The way that a G protein coupled receptor works is that it binds the ligand, shown here in red, and upon ligand binding, what it does is it activates a G protein found within the cell near the plasma membrane. So the G protein is a cytoplasmic protein that is located near the plasma membrane, so it is in the proximity of the G protein coupled receptor. When that G protein becomes activated, what it will typically do is it will then activate an enzyme. So there are many enzymes that are actually embedded in the plasma membrane or localized to the inner side of the plasma membrane within the cytoplasm, and G proteins can serve as molecules that can actually activate those enzymes, and that will ultimately lead to the catalysis of some sort of chemical reaction within the cell that will trigger further signal transduction pathways within the cell. So that's the basic workings of a G protein coupled receptor, but what may still be a little bit confusing here is what exactly this G protein is. And a G protein you can think of as a molecular switch, meaning that the protein itself exists in 2 different states, kind of like a light switch does. We have an on state and an off state. The G protein is in the on state when it is bound to GTP. Okay, GTP is an energy molecule that is similar to ATP that is used in the cell. And when the G protein is bound to GTP, the G protein is active. And so as we saw in the last slide, when the G protein is active, it could then go on and activate other proteins such as enzymes. However, when the G protein is bound to GDP, similar to the lower energy molecule ADP found within the cell, that puts the G protein into an inactive or off state. And the G protein will basically cycle between these 2 states, this on state and off state, or active and inactive state. In converting from the active to the inactive state, the G protein will undergo a GTP hydrolysis reaction whereby a phosphate group will be removed from the GTP, converting it to GDP, and that will essentially inactivate the G protein, shutting the signaling off. However, in the conversion from the inactive to the active state, an exchange reaction will occur. In this case, the GDP will be released from the G protein, and a GTP will come in to replace it and will bind to the G protein. So there is a GTP-GDP exchange that occurs, and when GTP binding takes place, that will activate the G protein. And then the G protein can go on and perform its signaling. The final type of receptor are the enzyme-coupled receptors. Enzyme-coupled receptors either act as enzymes themselves, or they associate with enzymes, and therefore lead to the indirect activation of an enzyme. And you'll remember that enzymes catalyze reactions that participate in many different intracellular signaling pathways. And an important thing to keep in mind about enzymes and enzyme reactions is that by far, within the cell, the most common form of activation of a protein is by phosphorylation. When a protein gets phosphorylated, it becomes activated. And so the act of phosphorylating or dephosphorylating a protein also behaves kind of like a molecular switch, where we have an on state or an active state and an off state or an inactive state for a particular protein. And in this case, the on state for the protein would be when the protein is phosphorylated or has bound a phosphate group. And the protein will be inactive when it does not bind to or is not phosphorylated. The type of enzyme that catalyzes phosphorylation reactions is a kinase. Kinases phosphorylate proteins. And the way that they do this is through ATP hydrolysis. ATP becomes hydrolyzed, and the phosphate group that gets removed from the ATP in the hydrolysis to ADP then gets transferred to the substrate. That will ultimately lead to the phosphorylation and typically activation of the protein. On the flip side, phosphatases are a family of enzymes that inactivate proteins. Phosphatases remove phosphate groups from proteins and therefore dephosphorylate the protein and typically lead to their inactivation. Many of the enzyme-coupled receptors fall into the category of what we call receptor tyrosine kinases. So what this means is that the receptor itself is a kinase. It is an enzyme that can phosphorylate substrates. And in the case of a receptor tyrosine kinase, in particular, the kinase activity phosphorylates tyrosine residues or tyrosine amino acids within the substrate. This is how a receptor tyrosine kinase works in very general terms. Here we're showing two receptor tyrosine kinases in the plasma membrane that have bound their ligand. When this occurs, when the ligand binds, it causes two of the receptors to closely associate with each other. So they move next to each other within the plasma membrane. And you'll recall that that plasma membrane is fluid and does allow proteins to move laterally within the membrane. So these two proteins will come into close proximity with each other, and this ligand binding facilitates this process that we call dimerization. A dimer is a group of two of something, so here we have two receptor tyrosine kinases in close contact with each other. When the receptor tyrosine kinases dimerize, that allows them to become kinases and to actually phosphorylate substrates. And the first substrate that they phosphorylate is each other. So these receptor tyrosine kinases actually phosphorylate the intracellular portion of the other receptor in the dimer. So you can see by my little messy arrows here, the left-hand receptor will phosphorylate the right-hand receptor, and the right-hand receptor will phosphorylate the left-hand receptor. And again, they're phosphorylating tyrosine amino acids that are found in that intracellular region of the receptor. Once the receptors themselves are phosphorylated, that further activates the receptor. That's through a process we call transphosphorylation, because we are phosphorylating the two different receptors within the dimer. And once those receptors are phosphorylated, that triggers the phosphorylation of intracellular cytoplasmic proteins that will continue to transmit this signal from the receptor. And again, the phosphorylation leads to the activation of an intracellular protein that can then lead to the phosphorylation and activation of another protein within the cell. That can lead to the phosphorylation and the activation of another protein within the cell. And again, these are a series of kinase reactions that are occurring, or phosphorylation reactions. And we call these reactions that occur consecutively a phosphorylation cascade or a kinase cascade, where one protein phosphorylates the next, which phosphorylates the next, etc., etc., etc. And that will ultimately lead to the activation of some sort of effector protein that will then alter the behavior of the cell. And so I want you to keep in mind receptor tyrosine kinases, because the paper that we'll be discussing in class on Wednesday is about a particular receptor tyrosine kinase called EGFR, or epidermal growth factor receptor. And this is a receptor tyrosine kinase that is often activated by mutations that occur in cancer. So it's constantly turned on, basically, and constantly sending signals to the cell that tell the cell to grow and divide, leading to cancer. And so we'll be examining the function of this EGF receptor in cancer in the paper on Wednesday.